Okay, hi guys. So my name is Max. I'm one of the SIs for Dr. Maduro's 5A section, and I'm just going to go over a couple of questions about nucleotides today. So these questions, they're either drawn from Professor Maduro's midterms, or the practice midterms rather, or they're very reflective of kind of the questions that we ask you guys on our SI worksheets. So go ahead and check those out if you want a bit more practice. So we'll go ahead and start with this first one on the left column. So the question is, the DNA molecule has the given sequence, and we're trying to figure out what the base pair molecule would look like, right? So a couple things to remember. First, you have to remember that kind of how DNA works, right? So DNA is anti-parallel, right? So what does that mean? That tells us that since our strand is 5' to 3', our kind of sequence that we're going to figure out will be 3' to 5', right? So that's just saying how in one direction it's going from 5 to 3, in the other direction it's going from 3 to 5, right? So think of it as like a two-way street. The second thing that we need to remember is what binds to what in terms of the base pairs, right? So we have adenine to thymine, and let me write these out, adenine to thymine. We also have cytosine to guanine, right? And since we're dealing with DNA, we don't have uracil, but if we did have uracil, it would be adenine to uracil, right? And uracil would take the place of thymine in this case. All right, cool. So with that in mind, let's go ahead and get started with drawing it. So if this is our sequence, I'm going to write it out again here. G A A T C C A 3', right? So that's our sequence, and we're trying to figure out what's kind of corresponding to that, right? You might think, so first of all, we might first probably label one side as 3' and the other side as 5'. That way we know that we're in an anti-parallel state. And now we look at the different base pairs, right? So and I'm gonna number these just so we have the same idea if I need to refer to specific bases themselves. All right, so if I'm looking at the first base, that's a G, right? So what binds with G, right? We said it was cytosine, so that's gonna be a C. And if we just follow this, what binds with A, that's gonna be a T, right? And then again, what binds with an A, that's again gonna be a T. And then what binds with T, that's an A, right? So these things aren't just A to T only, it can also be T to A, and so these all work. And so what binds with C, we did it earlier with the base 1, so that's a G. With 6, that's also a base, similar to the first base that we did, that's also a G. And then lastly, we have A, or adenine, and that's going to bind to a thymine. All right, cool. So we've done our complementary strand. So this is good for now, and it's good to know this, right? So this would be like a good answer, right? So at least we know now what the strand is. The thing is, they probably will give you the answers on your midterm from 5 to 3 as well, right? So our answer is going to come out as 5 to 3, and you might get a bit confused. You might say like, wait, isn't it supposed to be 3 to 5? And yes, it is, but in the answer choices, it's a way to add like a little bit more difficulty just to let you know, like, we're not just copying the strand again. Okay, so what's happening here is now I'm gonna number these again, but I'll use different colors. And we'll see how everything kind of changes. Okay, so you might already be thinking what we're gonna do here. In theory, we're not changing anything. We're simply flipping the order of it, and it's gonna be the same strand, right? So at our 5' end, we have our nucleotide 7, So that's a T, and then we have two G's, right? And we have an A, then we have two T's, oops, and then we have a C, right? And so if I numbered these again, you'll see how we kind of just flip the order of everything. We go from 7, 6, 5, 4, 3, 2, and then 1, right? So we just flip the order of everything. And so our final answer would be in this form right here, and I'll circle it in a brighter red. So this would be our final answer. But granted, both of these are correct, but I believe most of the answer choices would come out as a 5'. All right, so let's go ahead to the second one. So the second question is, what is a base pair, essentially, right? So we're trying to think about, how do we know something is a base pair? How do we know something is, another part of the nucleotide or DNA? All right, so let's look at this. So the first thing, so phosphate groups. So let's try to fill in the sentence. A base pair is formed by phosphate groups when they hydrogen bond, right? So think about where phosphate groups are and what they look like. So can they hydrogen bond? For one, they possibly could, right? We have a phosphate PO. In the case of a nucleotide, it's going to be a PO3 because one of the O's is going to be connected to the sugar. Which I'm going to just go right here. Sugar, right? So can it hydrogen bond? In this case, not really, just because it doesn't have an OH. If there was an OH, for example, if there was an H right here, then there would be potential for it to hydrogen bond. But since we don't have the OH, we can't hydrogen bond for one. And two, the phosphate groups, these are meant to connect, right? So think about what they're supposed to do. Phosphate groups, they connect the strands, they connect the nucleotides together, right? So the connecting of nucleotides, that's not a base pair. That's an actual covalent bond, and that's the backbone of DNA, right? So that doesn't work. I'm going to put an X through that in bright red. Dark red again. Okay, next thing. So we have when nucleotides interact with phosphodiester bonds, right? So this is similar to the previous point with phosphate groups. So nucleotides, that's when you have like your ribose or deoxyribose, right? You have your 5-carbon or 5-membered sugar. And then you would have the phosphodiester bands being, right, this oxygen here, and then you would be connected to a phosphate at that point, right? And then this phosphate would connect to an oxygen, like down here, for example. This would have a phosphate that would be also connected to like a neighboring that would be from a neighboring nucleotide, right? So that's the phosphodiester bond. So is that a hydrogen? Is that a base pair? Not really, again, because phosphodiester bonds, these are the phosphate groups that connect nucleotides, right? So phosphodiester, these are, I'm gonna label as nucleotide connecting or connectors. Okay, all right. Next thing we have is a base pair in DNA is formed by 2 ATP through hydrogen bonds. Okay, so ATP, that's a molecule you don't necessarily need to know the structure of. ATP does stand for adenosine triphosphate. Let me write that out. Right, ATP, hence the adenosine triphosphate, hence the ATP, so ATP right there. So that that's not really part of DNA, right? So you could have adenosine, which is definitely a precursor. It's like an adenine with 3 phosphates on it. However, DNA is not only made of adenines, for one, right? We have other bases that we talked about. We have thymine, cytosine, guanine. And so this doesn't really apply in this case, okay? All right, so I forgot to do so, but I'm gonna X through these two. So next we have deoxyriboses and glycosidic bonds, right? So deoxyriboses, that's when we have anything to do with DNA, right? So we're thinking, okay, maybe this is the one. Deoxyribose, DNA, D for deoxyribose. Glycosidic bonds, remember what those are for, right? That's when we have linkages between sugar molecules, right? So we might have an O connected to the rest of the deoxyribose, right? So that's a glycosidic bond right here. I'm going to highlight it in blue, this being a glycosidic bond. So it doesn't really signify a DNA connection, right? And we know that because glycosidic bonds connect sugars. But we kind of discussed earlier too that it's the phosphodiester bonds that connect nucleotides, right? So that leaves us with the last choice. And by process of elimination, this is going to be the correct one, but let's go over why. So the nitrogenous bases, that's when we have, you know, anything to do with nitrogenous. These are adenine, right, or A, T, C, G, any of those or U. But since we're dealing with DNA, we would only have ATCG. Okay, and so right now we're looking good. What does this non-covalent bond mean? Right. So this non-covalent bond, this is like an intermolecular force. So are we thinking that the bases interact by dipole-dipole interactions, by van der Waals interactions, or by hydrogen bonding? And so as you guys might know already, the nitrogenous bases, they have hydrogen bonding between them. And that's what leads them to getting interacted, and that's how we make the double-stranded DNA molecule, right? So this would be the correct answer for sure. Right, cool. So let's move on to the last one. So the last one is, what are the three main parts of a nucleotide and what makes them different from each other? Okay, so let's think about a nucleotide. I'm gonna draw a very crude one right here. I'm gonna color code it as well. So I'm gonna go blue. I'm gonna put this as just a B. And then go, oh. Okay, so ignore the crudeness of this drawing. Remember that we have three main parts, right? So we have the thing in red. So what's this thing in red? In maroon, we have our ribose or deoxyribose, right? So this is our pentose sugar, meaning something with 5 carbons or a 5-membered ring. Pent being the 5 and os signifying that it's a sugar. What else do we have? So we have our base, right? In blue. So that's our nitrogenous base. Right, so this is again our A's, our T's, C's, and G's. Okay, and lastly we have the phosphate that I put in pink. Phosphate, and this again will make up the phosphodiester bonds to make the backbone of DNA, right? And so all these three things together kind of, they make up the nucleotide. And so remember, for one, that between ribose and deoxyribose, we have 2 OH molecules right here. There are more OH molecules in this sugar, but I just didn't draw them in. We do have 2 OH molecules right here, but remember that the difference between ribose and deoxyribose occurs at this one, which we label as the carbon 2. Okay, so that's where the difference between ribose and deoxyribose is. The next thing is what makes them different. So you'll notice that every nucleotide has, even nucleosides rather, have the same kind of backbone, right? I'm going to highlight this backbone in green. So this backbone is always the phosphate and the sugar. Okay, so you'll see this kind of repeating unit where this phosphate will actually bind to the 3' carbon of another sugar molecule, right? And then this will have its own phosphate. And so they all kind of, this all repeats, right? So you can see that this repeating unit is happening. What's different is the base. So I'm going to write that in maroon again, but I'm going to write it up here. So the base make nucleotides different if you wanted to classify them as that, just because they kind of determine what's going to happen, right? An adenine is different from a thymine, not only because adenine is a purine and thymine is a pyrimidine, but also because of the properties of how many hydrogen bonds you're going to form, or things of that nature. So I hope this helped. If you have any other questions, please feel free to reach out to any of the SIs. Good luck on your midterm, and we hope to see you in session.